Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Has you ever found yourself wondering if seemingly different health challenges, like maybe the cognitive fog that cancer survivors often experience, or even just the memory issues that can sometimes come with aging, if they might actually share some kind of surprising common root? It's a pretty striking thought, isn't it? This cognitive fog, people call it chemobrain. It's a really debilitating side effect for a lot of people. And, well, it shows some truly fascinating parallels with the memory and learning challenges we often just chalk up to getting older. Welcome to the Deep Dive. Today we're digging into a stack of really interesting recent research from the University of Oklahoma. We've got some major scientific papers, a health news article, and we're going to explore this exact overlap. Our mission really is to unpack the shared mechanisms of cognitive decline in both chemobrain and the aging brain, and maybe reveal some groundbreaking research into potential solutions that could help with both. Let's dive in.
Yeah, it's a really compelling area because it connects these two conditions that, on the surface, they seem totally distinct. Chemobrain, that's the term most people know for the cognitive issues, right? Things like problems with memory, focus, learning, stuff that can unfortunately stick around long after chemotherapy. And even though chemo is obviously a lifesaver, these cognitive side effects can really impact a person's quality of life for, well, years sometimes.
Right, a serious challenge for survivors.
Yeah.
But you mentioned this surprising link to aging. What were the key things that Dr. Anna Cesar, she's the lead researcher from Oklahoma, right? What did her team observe that shows how similar these conditions actually are?
Well, Dr. Scissor and her team pinpointed three really significant parallels. First off, in both chemobrain and the aging brain, there's this noticeable drop in blood flow when the brain is just resting. But maybe even more importantly, there's a smaller increase in blood flow even when the brain is actively working hard.
Huh.
It's like, imagine your brain trying to run a marathon, but the fuel supply just can't keep up, It never quite gets enough.
Okay, that sounds pretty fundamental. A basic problem with fuel delivery. What was the second similarity?
The second one is all about the brain's protective shield, the blood-brain barrier. This is that highly selective filter. Its job is to keep nasty stuff out of the brain while letting the good stuff, like nutrients, in.
Right.
Well, in both chemobrain and aging, this protective layer gets compromised, it gets kind of leaky, and this disruption, this leakiness, it then triggers inflammation inside the brain.
Okay, like a security breach almost.
Exactly, like the brain's security system is down, letting troublemakers stir things up.
So a dodgy fuel line and a breached security system. And the third parallel, that's the one people often describe really vividly, isn't it? The zombie cells.
That's the one.
Yeah.
Yeah, the third really intriguing parallel is this buildup of what researchers often call zombie cells. The more formal term is senescent cells. These are cells that have basically retired, they've stopped dividing, but crucially, they haven't died off. Instead, they just hang around, they're still metabolically active, but unfortunately, they actively pump out these inflammatory substances that damage healthy cells nearby.
Wow.
Yeah, like grumpy old cells just making things miserable for the whole neighborhood.
Okay, let's unpack that a bit. We know chemotherapy drugs, they're designed to kill rapidly dividing cancer cells. But here's the puzzle. How do these drugs, which often don't even cross that protective brain barrier very easily, still manage to cause this kind of harm inside the brain?
That's a great question, and it really gets to the heart of this vascular connection we're talking about. The research suggests that chemo drugs don't necessarily need to cross the blood-brain barrier in large amounts. Often they're just too big chemically, or they don't have the right properties to slip through easily. Plus, the brain has ways to pump things back out. But they still harm the brain, just more indirectly. Their main hit seems to be on the endothelial cells.
Okay.
Those are the specialized cells lining all our blood vessels, including the really tiny ones in the brain. And they seem particularly vulnerable to chemo damage. When these endothelial cells get impaired, they become senescent, our zombie cells, and it's the inflammatory junk they release that then messes up the blood-brain barrier.
I see. So it's like a chain reaction. The chemo hits the vessel lining, that damage creates these inflammatory zombie cells, and they cause the barrier to leak, even if the drug itself didn't get in much. I saw one study mentioned focusing on paclitaxel. That's a really common chemo drug. What did that specific study find?
Yeah, that was a big study in geroscience looking at paclitaxel in mice. And the findings were quite eye-opening. They found that even though different chemo drugs might damage DNA in different ways, their effects on cognition seemed pretty similar. What the study really showed was how chemo can physically change the brain's microvasculature, its tiny blood vessels. With paclitaxel, they saw a significant and long-lasting reduction in the density of these small blood vessels. It's called microvascular rarefaction.
Microvascular
Right,
rarefaction.
especially in the cerebral cortex and the hippocampus, key areas for learning and memory.
Wow.
It's like the brain's delicate irrigation system gets pruned back, basically starving those vital areas.
So a literal loss of these tiny, crucial blood vessels. And did that affect the bigger picture, like overall blood flow?
Oh, absolutely.
Yeah.
The study also showed a significant drop in the speed of blood flow in the larger arteries supplying the brain, like the middle cerebral arteries, so
Yeah.
impaired brain perfusion overall. Think of it like a slower delivery service for everything the brain needs, and maybe even more worrying. They saw significantly impaired neurovascular coupling.
So vascular coupling, what's that exactly?
It's the brain's amazing ability to boost blood flow to specific areas that become active, like when you're concentrating hard or learning something new.
Right, sending resources where they're needed most.
Exactly. Paclitaxel treatment really dampened this response in the mice. Less blood flow right when the brain was demanding it.
That paints a pretty stark picture for paclitaxel, widespread vascular problems. What about other common chemo drugs? I think cisplatin was mentioned. Did they find similar effects on the blood-brain barrier there?
Yes, they did. A separate pilot study looked at cisplatin, another very widely used chemo drug, and it confirmed this persistent disruption of the blood-brain barrier. They used some advanced imaging, basically injecting different size glowing molecules.
Like tracers?
Exactly, tracers, to see how easily they leaked across the barrier. They saw significantly more leakiness in the cisplatin-treated mice, and it lasted for up to 2 months after the treatment stopped. What was really insightful was that the smallest tracer leaked the most, which strongly suggests that cisplatin primarily messes with the seams between the endothelial cells, the sort of tight junctions that normally keep the barrier sealed, making it easier for things to slip through between the cells.
Okay, so we've got these senescent zombie cells popping up, causing inflammation, making the barrier leaky. But why are these cells such a big driver? And do we know where they tend to show up first in the brain?
Well, what's really fascinating and kind of central to this whole idea is that senescent cells aren't just a side effect. They seem to be key drivers of the problem. Remember, they've stopped dividing, but they're actively churning out inflammatory factors that spread damage around them. It really is like that bad apple spoiling the barrel, but on a cellular level. And a really crucial insight came from another study, this one in aging cell. It found that the brain's endothelial cells, those same blood vessel lining cells we talked about, seem especially sensitive to aging. They become senescent faster and earlier than other brain cell types, particularly starting around middle age.
Middle age.
Yeah, in mice, this was seen around 15 to 17 months old, which the researchers think roughly corresponds to humans in their 50s.
Wow, that is significant.
Huh.
It suggests that the aging of our blood vessels, the vascular aging, might actually come before major problems with the neurons themselves. Like the supply lines start failing before the brain cells really suffer. That's kind of counterintuitive, isn't it?
It really is. Many might assume it's the neurons that go first.
So how does this breakdown in the vessels affect how cells talk to each other in the brain?
Well, connecting the dots, researchers use some sophisticated techniques like single-cell RNA sequencing to look at communication between cells. And they found a general decline in cell-to-cell chatter as the brain aged. Specifically, weaker communication between the endothelial cells, parasites, which are like support cells wrapped around capillaries and astrocytes, another key support cell.
That sounds like the whole support crew for the blood vessels is breaking down.
Pretty much. This team, called the neurovascular unit, is crucial for a healthy brain environment. What they saw was less of the good signals, things that promote blood vessel health and growth, and more of the bad inflammatory signals. So it's a double whammy, really. Less support, more damage.
Okay, this is all sounding, well, pretty concerning. It makes both chemobrain and age-related decline seem quite daunting. But you mentioned a ray of hope earlier, these things called Senolytic therapies. What exactly are those? How do they work?
Yes, absolutely. There is definitely hope here. Senolytics are a really revolutionary type of drug. They're designed to selectively trigger these senescent cells, the zombie cells, to undergo apoptosis.
Apoptosis.
Yeah.
That's programmed cell death, right?
Exactly. It's the body's natural way of removing old or damaged cells. So Senolytics act like a highly targeted cleanup crew. They go in, identify just the problematic senescent cells, and tell them it's time to go, leaving the healthy cells alone.
The targeted cleanup crew.
Yeah.
I like that. And what happened when they tested these in those aging mice studies? Did it actually help reverse some of these issues?
The results were remarkably promising, actually. In the aging cell study, they tested Senolytics compounds like ganciclovir and another called navitoclax in aging mice. And by removing those senescent cells, they saw significant improvements across the board.
Like what?
Well, better cerebral blood flow, for one. The blood-brain barrier started to heal, its integrity was restored. And remarkably, these benefits lasted for up to 6 months after just one round of treatment.
6 months from one treatment.
Yeah, pretty amazing. They also saw increased density of those tiny microvessels in the brain's cortex, and better neurovascular coupling the brain's ability to direct blood flow improved. And cognitively. The mice got better at spatial learning tasks like navigating mazes. They made fewer errors.
Wow.
Now, it didn't fix every aspect of cognitive flexibility they tested, but the overall improvement was really quite profound.
That's incredible, especially the lasting effects from a single treatment. But that brings up a question. Does it matter when you give these drugs? Is there an optimal time window for them to work best?
That seems to be a really critical factor. Timing. The research suggested the drugs worked best when given to the mice around 16 months old.
Which is like human middle age, 50s-ish?
Right. What the researchers estimate corresponds to humans aged about 50 to 55. What's fascinating, and maybe a bit sobering, is that while the senolytics could still clear out senescent cells and protect the blood vessels even if given later, the cognitive benefits weren't as pronounced. It seems some cognitive changes might become irreversible if you wait too long. So it really highlights the potential importance of acting earlier, maybe proactively, before significant cognitive decline really takes hold.
Okay, so these findings are undeniably exciting. A potential way to target these zombie cells and improve vascular health, maybe even cognition. But we have to acknowledge these studies were mostly in mice, right? What does that mean for applying this directly to people?
That's the crucial caveat, yes. While the results are super compelling, these were mouse studies. Now, researchers have looked at human brain tissue and found similar markers of senescence, which is encouraging. It suggests the parallels are real, but moving from the lab bench to actual human treatments, that requires a lot more rigorous research. Clinical trials in people are essential. It's a big leap.
And were the studies totally comprehensive? Did they always look at cognition alongside the vascular stuff, or check all the different cell types involved?
Not always, no. Some studies understandably focused more heavily on the vascular outcomes, the blood flow, the barrier integrity, and they didn't always include direct cognitive tests or analyze senescence in every single type of brain cell.
Okay.
So there's definitely room for future research to broaden the scope to get an even more complete picture of exactly who gets senescent and how it impacts everything else.
Right. And what about the mice themselves? You mentioned they used mice without tumors for the chemo studies. How might that affect how we interpret the results for actual cancer patients?
That's a very sharp point. Using non-tumor-bearing mice was a deliberate scientific choice. It helped them isolate the direct effects of the chemotherapy on the brain's vascular system without the cancer itself confusing things.
Excellent.
Because cancer can cause its own inflammation, tumors can release substances, lots of confounding factors. But you're right, the real-world clinical situation involves both the cancer and the treatment. So these studies give us a clean look at the chemo effect, but we need to remember the actual patient experience is more complex.
It definitely sounds like there's still plenty to figure out, but the foundation laid here is incredibly promising. What are some other key questions or areas that future research needs to tackle?
Well, one big area is looking at potential sex differences. Many of these initial studies focused mainly on male mice, so understanding if these processes affect males and females differently is important. And also, digging deeper into how chemotherapy affects other brain cell types beyond just those endothelial cells lining the vessels, getting that fuller picture of all the cellular players involved in chemobrain and aging.
Okay, so let's pull this together for everyone listening. We've explored this really striking overlap between chemobrain and normal cognitive aging, driven by these core problems. Less blood flow, a leaky blood-brain barrier, and these pesky zombie senescent cells causing inflammation. But then we also saw this exciting potential with senolytic therapies, drugs that might actually target and remove those problem cells, potentially reversing some of the damage.
Absolutely. I think the core takeaway message here is just how critical our microvascular health is for cognitive function, whether the challenge comes from chemotherapy or just the process of aging. The health of our tiniest blood vessels seems intimately tied to our cognitive destiny, you could say. This research really shifts the focus, suggesting maybe the fountain of youth for our brains isn't some magic potion, but rather lies in maintaining and perhaps repairing these delicate internal vascular systems.
So if the key to keeping our minds sharp, maybe even a sort of fountain of youth for the brain, lies in the health of our tiniest blood vessels and clearing out those cellular zombies, it leaves us with a thought, doesn't it? What small steps might you consider taking right now to support your own vascular health, knowing just how closely it's connected to your cognitive future? Something to think about.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.